have a person about to eat a delectable Danish pastry. But how does the body digest it? For that, the human body needs the protein Danishes to break down the Danish pastry. So how is Danishes made? First, in the nucleus, we find the genes on the DNA that code for Danishes. In that section, all we care about is the part that codes for Danishes. The other part, the antisense strand, we don't care about. As a side note, there are free-floating nucleotides in the nucleus. RNA polymerase Pac-Man goes in and unzips the section of DNA that codes for Danishes from the antisense strand. Then, with the transcriptional factors on it, begins to transcribe the DNA, coming out with mRNA. In this case, the transcriptional factors are red spheres of sugar that will represent the digestive properties of Danishes. Before RNA polymerase can start, it needs to have a place to start. The Tata box, which is a series of T's and A's, offers a place for RNA polymerase to start. To remember the Tata box, think of Tigger since his tail looks like a Tata box. Afterwards, RNA polymerase reaches the promoter, which signals the true start of transcription to mRNA. Think of a promoter as a giant signboard to start. Afterwards, the RNA polymerase stops transcribing when it reaches the termination sequence, which tells the RNA polymerase to halt, since the gene could code for multiple proteins that have many codons. Think of Arnold Schwarzenegger as the terminator at the end of the DNA. After the termination sequence, the completed mRNA undergoes RNA processing, in which it removes introns. Introns are sections of DNA on a gene that do not code for anything. SNRP, the signal recognition particle, pinches introns out. SNRP then splices together exons, which are the parts that code for something. To remember this, think of SNRP as scissors. To remember introns and exons, introns stay in the nucleus, and exons exit the nucleus. In around the same time, a 5' cap consisting of PPP and G and a poly A tail attached to the mRNA, ensuring that it has a safe passage from the nucleus to the ribosome. Think of them as bodyguards so that nothing can harm the mRNA. The mRNA then leaves the nucleus via the nuclear pores and heads to the ribosome. The ribosome has 2 layers and the mRNA goes in between them. The top layer is further divided into 3 chambers, the A, P, and E. To remember this, think ape. The mRNA first goes into A, the acceptor chamber. There, tRNA has an anticodon, which is complementary to the codon. The tRNA finds the free-floating amino acid that correlates with that codon. In this case, it codes for MET, which is the start codon. It then shifts over so that the next codon is read and has its subsequent amino acid brought. In the P, the peptidal chamber, amino acid tRNA synthetase makes peptide bonds between the amino acids. The amino acids also disconnect from the tRNA at this point. Again, mRNA shifts over repeatedly until it reaches the stop codon. At the E, the exit chamber, the tRNA leaves the ribosome and continues to look for amino acids again. Also at the E, the mRNA loses its bodyguards, the 5' cap and the poly A tail, which lets it be degraded by other enzymes in the cell, thus preventing an overpopulation of mRNA in the cell. The completed polypeptide chain also leaves at E. And there you have it! We now have the enzyme Danishes that will allow this student to digest his Danish pastry. Hope everyone learned a lot, and good luck on finals. Not that you guys need it because you're all so smart and my favorite people ever. I love you!